All right, so thank you for being here, everybody. My name is Patty Jones. I'm the Associate Director for Research here at the Beckman Institute, and it's my pleasure today to welcome all of you to the February 2021 Beckman Institute Directors Seminar. This seminar series has existed ever since the Beckman Institute was founded, and it is a way for faculty members who are members of Beckman to give a talk to a fairly general audience across all of the different Beckman research themes. So today we are really pleased to welcome, as you can see on your screen, Professor Catherine Christian Hinman from the Department of Molecular and Integrative Physiology. Her background is in neuroscience, where she received her Ph.D. in neuroscience from the University of Virginia, and she completed her postdoc training in neurological sciences at the Stanford School of Medicine. And again, as you can see on the screen, she's currently an assistant professor of molecular and integrative physiology here at U of I. She's also a faculty affiliate of the Beckman Institute and of the neuroscience program. And as we will hear more about today, her current research focuses on neural mechanisms linking epilepsy and comorbid reproductive endocrine disorders and interactions between seizure activity and the reproductive cycle. And her research is obviously very interdisciplinary, looking at a variety of methods. For this seminar, what we're going to do is everybody's muted, so Catherine will just give her talk straight through. And then at the end of it, we will unmute your microphones, and so you are able to ask questions in person. But also during Catherine's talk, please feel free to use the chat, which I'll be monitoring, so I can also harvest questions at the end of the talk from the chat box as well. So with that, let us all give a warm virtual welcome to Professor Christian Hinman. Thank you for being here.
Great, thanks, Patty. And I'd like to thank you and Stacy and, of course, Jeff Moore, kind of in absentia. I knew that he couldn't make it today, but for the invitation to present some of my lab's work in the Beckman seminar. I'm very honored to be offered this slot. And so just to get started, so as Patty mentioned, a large focus of my lab's research is in the field of epilepsy, and which is characterized by seizures. And a seizure is a brief and strong surge of electrical activity in the brain, but there are many different types of seizures, and there are many different causes of seizures. So for example, we can see seizures that stay primarily confined to one part of the brain, in which case we would say that that's a focal seizure. But you can also see seizures that have a very different presentation, where they exhibit generalized synchronous activity, typically throughout the cortex. And so you can also have seizures that start focal and then secondarily generalize. So seizures themselves are heterogeneous, and there are many different causes of seizures, and this will also have many different presentations for different patients in terms of the types of symptoms that they show and experience. However, having just one seizure does not mean that someone has epilepsy. The clinical definition of epilepsy is spontaneous and recurrent seizures over time. And what we mean by spontaneous are unprovoked seizures. So again, a seizure could be elicited by many different acute causes, but someone who has epilepsy will have these episodes periodically and typically unpredictably for large portions of their life, especially if the seizures are difficult to treat. And to give you some statistics about the public health prevalence of epilepsy and why it's so important for us to learn more about it, unfortunately, the number of people with epilepsy keeps growing. It affects approximately 1.2% of the population, including a large number of children as well. I'll be focusing primarily on a form of epilepsy that's seen primarily in adults. But again, this is something that is seen throughout the world in all socioeconomic classes. And it really is a growing public health problem. And especially because of the heterogeneity, that makes it even more difficult to treat. And it sort of leads us to really want to think a little bit more outside the box as ways to identify new therapies and new potential targets for treatment. So as I mentioned, epilepsy itself is sort of a heterogeneous disorder, and we would actually even really say that it's more of an umbrella term, that there are multiple epilepsies. And the form that my lab is focused on is called temporal lobe epilepsy, and I'll be abbreviating this as TLE. This is the most common form of a focal epilepsy that's seen in adults, and it's often characterized by lesions or sclerosis of limbic structures such as the hippocampus or the amygdala. Now, again, even within TLE, we see heterogeneity in this type of histopathology. And this is shown in this example figure on the right, showing staining through a section of hippocampus obtained from a person that did not have epilepsy, and then comparing this to 3 different types of hippocampal sclerosis that were all seen in patients that had temporal lobe epilepsy, used as a general term. So again, even within one form of epilepsy, we see many different types of epilepsy and type of cellular pathology. And again, even within TLE, there are many different causes of TLE. It typically arises after some form of a severe insult. So for example, a traumatic brain injury or stroke in older populations or adults, but also febrile seizures, which are seen in toddlers during a bout of high fever. And that provokes a strong bout of seizures that often remit, and then that child doesn't show seizures for the rest of their childhood. But as they get into late adolescence and adulthood, they start exhibiting temporal lobe seizures. However, there are also genetic factors as well, and we keep learning about more and more gene mutations that are linked to forms of altered neural activity and seizure activity. Unfortunately, TLE is also the most common form of a medically intractable epilepsy, and approximately 1/3 of patients with TLE don't respond to current drug treatments. And so again, it really underscores the need to identify new potential targets for treatment and new avenues of investigation. My lab is focused in a particular aspect of temporal lobe epilepsy, and that's the fact that it appears to be linked with much higher rates of reproductive endocrine comorbidities. And this is seen in both women and men, but as you can imagine, the exact presentations do vary with gender. So in women with TLE, we see higher rates of altered menstrual cyclicity, polycystic ovary syndrome or PCOS, or at least some features of the syndrome, such as polycystic ovaries on their own. Hypothalamic amenorrhea, which means a disruption to the menstrual cycle due to disruption at the hypothalamic level within the brain. And you'll see that I'll be focusing on that particular aspect later in the talk. Hyperandrogenism, or high circulating androgen levels, and also early menopause. And whereas in men, we see higher rates of hypogonadism, abnormalities in semen, and low testosterone. And so to give you some idea of the statistics of how patients with TLE exhibit these higher rates of reproductive problems, polycystic ovary syndrome, just use this as an example, is the most common form of a fertility disorder seen in women of the general population, affecting approximately 5 to, depending on the study, sometimes 10% of women of the general population. But if we look at women with epilepsy, we see that that rate increases to approximately 20%. And then if we look at menstrual disorders more broadly and focus on women with TLE that are not taking anti-seizure drugs, so these disorders would not be a result of side effects of anti-seizure drug treatment, we see that approximately 60% of these women show some form of a reproductive comorbidity. So this is certainly something that can significantly impact quality of life. And also general endocrine health really is sort of a window to overall health. And so it's not just about the epilepsy itself, but these other symptoms that these patients experience that can influence their overall quality of life and potentially also influence the epilepsy itself. And the reason that I say that is that we know that there are very interesting relationships between the reproductive system and the brain overall, and certainly how this manifests in the context of epilepsy is a very important problem. So if we look in women with epilepsy that are able to maintain normal menstrual cycles, we see that there are often times when women show increased seizure susceptibility and frequency in relation to different phases of the menstrual cycle. So one of these periods is near the time of ovulation, near the midpoint of the menstrual cycle, so which is depicted here. So starting at day 0, the onset of menstruation, through a typical 28-day cycle. And you see that leading up to the time of ovulation, which occurs at the midpoint of the cycle, there's a rise in estradiol, which is depicted in the blue line here. And this is thought to not only be necessary to trigger ovulation, which we know that it is, but also when this estradiol hits the brain, it's thought to have primarily proconvulsant or excitatory activity, so effects that provoke seizure activity. However, another population of women with epilepsy will show increased frequency of seizures near the time of menstruation, or certainly in the few days leading up to menstruation. And this is thought to be primarily precipitated by this drop in progesterone, which is depicted in the red line here. And the reason that it's thought that progesterone may play a role in this particular relationship is that when progesterone reaches the brain, it's converted to substances called neurosteroids, which help to potentiate inhibitory signaling in the brain and decrease neural excitability and decrease the propensity for seizures. So overall, both of these phenomena are referred to as catamenial seizure clustering, or increased seizures at certain points of the menstrual cycle. But we've been particularly interested in another form of catamenial epilepsy in which women have disrupted ovulation and therefore disrupted menstrual cycles. And because these women don't ovulate, they still experience the same pre-ovulatory rise in estradiol, but because the ovary does not release an egg and you don't get the same formation of the corpus luteum in the ovary, which is what produces progesterone, you don't get that same post-ovulatory luteal phase rise in progesterone. And so now we have this sort of milieu of increased estradiol, which again primarily has proconvulsant excitatory effects in the brain. And so these women have a much more prolonged window of increased seizure susceptibility. And so this indicates that it's not only an issue of the relationship of hormone levels and the menstrual cycle and seizures, but also if the reproductive axis is significantly impaired, that can also influence the epilepsy itself. And so I hope that you're starting to appreciate that we have this reciprocal feedback loop where we see that temporal lobe seizures can precipitate comorbid reproductive endocrine dysfunction. And then in turn, that dysfunction can also influence the presentation of these temporal lobe seizures. And in terms of what I'll be focusing on in this talk, my lab has been focused on this upper arm, trying to understand how temporal lobe seizures are linked with reproductive endocrine problems, specifically trying to elucidate the neural mechanisms within the brain that link these two physiological systems. So to do this, we focus on using a mouse model of temporal lobe epilepsy called the Intrahippocampal kainic acid, or IHKA model. This is a model that's been used in our field for about 25 years, in which the glutamate receptor agonist kainic acid is injected directly into the hippocampus. So the advantages of this are that we can precisely target the kainic acid injection to the hippocampus. And then especially for our particular research questions where we're interested in changes in downstream endocrine function and in changes in other parts of the brain that are not necessarily where the seizure activity is, we can be more confident that these changes we see later on are due to effects within the brain and not because a systemic treatment was given to induce the epilepsy, which is the other main model. And I will have one slide where I talk about another model of TLE. And so one of the reasons that this model is so widely used in the field is that it does recapitulate several features of human TLE, including the hippocampal cellular pathology of the sclerosis and gliosis, or proliferating glial cells in the hippocampus. And this is an example of a kainic acid injected hippocampus from a mouse compared to that of a saline injected control. And so you can see the dramatic difference after the kainic acid injection. And also, if we do EEG recordings or electroencephalography to record seizure activity in these animals, we see similar features in these recordings to what you can see in patients with TLE. And so overall, no model is perfect, but this model certainly does help us to be able to do some studies in animals that we couldn't do in people. And also in terms of whether it's appropriate for us to be studying this type of question in rodents, I do want to emphasize that, of course, rodents are not people. And so the female reproductive cycle in a rodent, here it's depicted from a rat, but that in the mouse is very similar, is called the estrus cycle. And again, you can see that there's a midpoint in the cycle at which ovulation occurs. And leading up to that, we see a rise in estradiol, similar to what we see in women before ovulation. There's also a rise in progesterone around the time of ovulation, but as you can see, it's of much shorter duration than what is seen in women. And that is really the main feature that I would say is very different between these two forms of cycles. But at least there are enough similarities that again, in terms of looking at these questions in rodents, we can start to ask questions and hopefully get some answers that would be very difficult to do in a clinical population. So our first question was whether the female mice in the IHKA model of temporal lobe epilepsy show altered estrus cycles. And so to do this, we use a method that's been around for over 100 years of daily vaginal smears. It's essentially a little bit like a pap smear for a mouse. So we inject a small volume of saline into the vaginal cavity and remove it. And when we remove that saline, that withdraws some very loosely held cells lining the vaginal wall, which we can then put on a microscope slide and look. And by looking through the scope, we can observe the changes in the number and the morphology of the cells. And when you do this day after day, you can start to see these changes in these cells, and you can classify the mice as being in one of these 5 stages of the cycle. So one day of proestrus, a day of estrus, metestrus, and then 1 or 2 days of diestrus. So in this first set of experiments, we took female mice that were adult and reproductively mature at 42 days of age and older. We did this daily cycle monitoring for at least 2 weeks to make sure that they had normal cycles before they were entered into the experiment. And then on what we designated as day 0, we injected either kainic acid or control saline into the hippocampus. And then we resumed the daily estrus cycle monitoring 2 weeks later and continued to do this daily monitoring for up to 2 months after the injection. And here, what we've plotted is the percentage of time the mice were spending in each of these cycle stages during the pre-injection period and then at different time points after the injection. And so you can see that in the saline injected controls, there was no significant change in the proportion of time spent in any of the cycle stages. But when we did the same analysis in mice that were injected with kainic acid, we saw that it took at least 6 weeks for us to start to see significant changes. And this was primarily characterized by an increase in the amount of time spent in diestrus and a decrease in the amount of time spent in estrus. And so coming to the summary data, you can also see that when we look at the proportion of mice showing irregular or regular cycles in the saline injected controls and the mice injected with kainic acid, we saw a large increase in the proportion of mice that showed irregular cycles in the kainic acid injected group. But we did have this subpopulation that was resilient, I guess we could say, and were able to maintain regular cycles. And so we're very interested in this because this allows us to have essentially a population that has epilepsy that was exposed to kainic acid, but some of the mice have the comorbidity and some don't. And so this is much more like what we see in the clinical population, where again, it's a proportion of women with epilepsy that will have reproductive cycle problems. And one of the main features of this irregularity was an increase in the cycle length or period. So the amount of time it takes the mice to progress through all of the stages. So again, in the controls, this typically takes place over about 5 days. But in the group that was injected with kainic acid, we saw that this time was almost doubled, but on average to about 8 to 9 days. And so you'll see that we use the cycle length as one of our main parameters going forward in terms of being able to classify mice as having a cycle that's disrupted or not. Now, we wanted to see whether the disruption to the cycle was reflected in changes in the ovaries. And so in collaboration with the histology core in the School of Vet, or the College of Vet Med, we performed thin sections through the ovaries and stained, and that allows us to identify different forms of follicles in the ovaries. In the mouse ovary, you'll always find follicles that are in different stages. So it's not that they're all in one stage. You'll always find sort of a mix. But overall, when we looked in the ovaries obtained from the saline injected mice and the kainic acid injected mice, overall, there were no changes in either any of the different follicle types or in the presence of corpor-lutea. And again, this is important because that's what's left over after ovulation. So it suggests that these mice are ovulating properly, by and large. But that leads us to think, well, again, the cycle's disrupted. So how is it that we could have a disrupted cycle in these mice that have epilepsy? So that brings me to the hypothalamic-pituitary-gonadal axis, or the HPG axis. And this is comprised of the gonadotropin-releasing hormone, or GnRH neurons, that are located in the hypothalamus, which is at the base of the brain. And these neurons release the GnRH hormone into the vasculature near the pituitary gland, so into the bloodstream, so that GnRH can reach cells in the anterior pituitary that produce the gonadotropin hormones, luteinizing hormone, or LH, and follicle stimulating hormone, or FSH. And then in both sexes, LH and FSH act on the gonads to stimulate the production of steroid hormones. So in the males, this results in the production of testosterone, and in the females, this results in the production of estrogen and progesterone. And all of these steroids feed back at both the pituitary and the brain levels of this axis to regulate the overall activity. And my lab has been focused on the changes that we see in the hypothalamus, and then as well also in the pituitary. I won't have time today to go into the work on the pituitary, but it's definitely something that we're very interested in exploring. And so we've been focusing on the GnRH neurons because, again, they're the main output from the brain. And the official definition is that they're the final common pathway in the central control of fertility for all mammalian species. And so basically what that means is that in all mammals, you'll see that GnRH neurons do this job of integrating all of these different inputs, both from other types of neurons as well as from glial cells, and synthesizing that and taking that information to say whether the GnRH hormone should be released into the pituitary vasculature to provide a signal to the rest of the HPG axis that this organism is in an appropriate physiological state for reproduction. GnRH neurons are located primarily in the hypothalamus, but what's also very interesting about them is that they're actually scattered throughout different portions of the hypothalamus, including an area that's technically not part of the hypothalamus, the medial septum, but most of the GnRH neurons are in the preoptic area and then into the mediobasal hypothalamus. And depending on the species, this particular scattered pattern can be a bit different. But again, all mammals show this scattered pattern of distribution of the GnRH neurons. And you can see this scattered distribution in this staining study that was from Ed Dudek's lab several years ago, where they were studying a different mouse model of TLE in which the cholinergic agonist pilocarpine is given systemically. And this is another very widely used model of TLE. But as I mentioned, this results from a systemic injection. And so for our purposes, we've decided not to focus on using it. But I did want to show you their finding here, because when they stained for the GnRH peptide and looked at different portions through the hypothalamus, so these figures are going from left to right, going from more anterior to more posterior, you can see that both in terms of the cell bodies of the GnRH neurons, as well as their axons that come together in this massive innervation at an area called the median eminence. So this is where the GnRH hormone is released. you can see that by and large, there really isn't a difference between the control mice and those that were treated with pilocarpine to develop epilepsy. And so this suggests that if there is an effect at the level of the GnRH neurons, it's not because that epilepsy is inducing massive cell death or major changes in the morphology of the GnRH neurons, but it leaves open the possibility that more subtle functional changes in the electrophysiological properties of these cells may be altered. And so that's what my lab wanted to address. And so to do these studies, we're using a mouse that expresses a red fluorescent protein called TLE in GnRH neurons. And so here you can see a distribution of these red cells that is very similar to what you saw on the previous slide for the staining for GnRH. And so what this allows us to do is to prepare brain slices through the hypothalamus and target individual cells for recording. So that's what we did in this next series of studies, where we looked first at changes in GnRH neuron firing activity. And all of these experiments were done 2 months after injection of either saline or kainate into the hippocampus. And again, the reason that we chose 2 months is that we had seen that before it took at least 6 weeks for changes in the estrous cycle to begin to present. So the way that we do these experiments is we prepare acute coronal brain slices through the basal hypothalamus of our GnRH-tdTomato mice. And then we can take a recording pipette and position it to target an individual red fluorescent cell. And first we used a recording configuration called loose patch or targeted extracellular recording, which allows us to record the action currents or the fast currents that underlie the action potential without breaking the cell membrane of the cell that we're recording from. So this is a relatively non-invasive method of recording, but because it's targeted to an individual cell, we can have confidence that the cell that we're recording from is the type of cell that we're interested in. And you'll see that we did do these experiments in both females and males. And for the females, we prepared slices and recorded the cells on either diestrus or estrus. And we chose these 2 different stages of the cycle because in previous work by other groups, it had been suggested that these stages may be linked to differing seizure susceptibilities. So we were interested to know whether first of all, GnRH neurons show altered firing activity in mice with epilepsy, but also whether that relationship changes with the estrous cycle. So first, going over the data obtained from cells recorded on diestrus, we found that mice that were injected with kainic acid and developed long disrupted cycles, which we defined as a cycle length of 7 days or longer, their GnRH neurons showed increased levels of firing compared to both cells obtained from saline injected controls, but also from, in comparison to cells obtained from kainic acid injected mice that were able to maintain regular cycles. So on the left, I'm showing the summary data for all of the cells recorded. And then on the right, we have the data for each of the individual cells. And you can see that there's quite a bit of heterogeneity and spread. And on the next slide, I'll come to at least one factor that we think may underlie this heterogeneity and explain it a little bit. So that's what we saw in diestrus, that again, the mice that have the comorbid estrous cycle disruption typically show increased activity at the level of the GnRH neurons. But then what we were rather very surprised to see was that when we did the exact same experiments, but now preparing slices and recording cells on estrus, we saw that the cells from the kainic acid injected mice showed the opposite relationship. So first, if we compared the cells from the saline injected controls and compared them to the cells that were recorded on diestrus, we saw that typically the cells that were recorded on estrus showed increased rates of firing compared to the corresponding cells recorded on diestrus. But in the case of the cells recorded from the kainic acid injected mice, we saw that that relationship was flipped. So now we saw strongly suppressed levels of firing. And we also saw this in both of the kainic acid groups. So whether or not the cycle length was long or regular. And so again, this is shown in the summary data on the left and in the individual cell data on the right, where again, you can see some subpopulations jumping out in the data. So as I mentioned, GnRH neurons have a scattered distribution. So we thought, well, perhaps this heterogeneity is related to the location of the cell within the hypothalamus. And indeed, that is what we found. So we took the exact same data, but now subdivided it based on whether the location of the cell body was in the medial septum or the MS, the preoptic area or POA, or the anterior hypothalamic area. And I'd like to emphasize that again, as we're going from left to right, we're going anterior to posterior. And so unfortunately here it has the word anterior, but it's actually the most posterior of the regions that we looked at. And what we found was that it was largely the cells in the medial septum and preoptic area that seemed to be affected by epilepsy in the kainic acid long group, whereas we didn't see any differences comparing the cells that were recorded within the AHA. And similarly, when we looked at the data obtained on estrus, again, we saw it was cells in the MS and the POA that showed strongly suppressed levels of firing. And then when we looked in the AHA, now we did see that the cells from the KA long group were also suppressed, but we didn't see that so much in the KA regular group. So it's not just a matter of sort of the functional heterogeneity, but it indicates that the location of the cell, and therefore also most likely the synaptic inputs that the cell is receiving, may also influence how these cells respond in the face of epilepsy. So that was what we'd seen at the level of the firing output of the cells. And so we wanted to understand whether this was also reflected in changes in the excitability or the propensity for these cells to fire action potentials. And so to answer this question, we turned to another recording configuration called whole cell current clamp recordings. And what this allows us to do is to have more electrical access to the cell, and we can inject differing amounts of depolarizing current and count the number of action potentials that are elicited. So in essence, the more action potentials that we see in response to the same stimulation, that means that that cell is more excitable. Now, here we have some example cells from saline injected controls and kainic acid injected mice recorded on both diestrus and estrus. And as you can see, we see more of these action potentials or spikes in both of the cells from the kainic acid injected mice, independent of the cycle stage. When we looked at the threshold for action potential firing, or the membrane potential at which an action potential is triggered, we saw that by and large there was a hyperpolarization or a movement of this threshold towards more negative values in the cells from the kainic acid injected mice. And again, this would be reflective of increased excitability, or that it's easier for these cells to fire an action potential. We did not see that this was reflected in changes in the input resistance of the cell. Again, now we're getting into some very specifics about cellular electrophysiology, but just for those of you that are interested in this, I did just want to point out that GnRH neurons are small, and so they have high input resistance to begin with, certainly if you compare this to your typical hippocampal cell, for example. And so it could be that we're just at a point where we can't really see that there's really an increase in the input resistance. But certainly when we looked at the number of action potentials elicited in response to these depolarizing stimuli, overall we saw an increase in excitability. And this is quantified here for the cells, again, broken down based on cell location in the medial septum, the preoptic area, or the AHA. And so first, to look on the left for the data recorded on diestrus, we saw more action potentials, or a higher evoked firing rate, across these increasing amounts of depolarizing current injection. And in fact, we saw this in some cases in both mice that had regular cycles, even though they were injected with kainic acid, as well as those that had long disrupted cycles. But again, when we looked in the AHA, we actually didn't see much of a phenotype here. And this appeared to be primarily reflected in the fact that within the saline injected controls, those cells were already a little bit excitable. So even within the controls, there's heterogeneity in these excitability properties. But then what was interesting was when we did these same recordings in slices prepared on estrus, we also saw increased excitability in the cells recorded from the medial septum or the preoptic area. And so now we have a little bit of a dichotomy, because we saw that on estrus, by and large, the cells, if we recorded the firing output that they were endogenously exhibiting, so without any sort of stimulation, we saw that their firing was strongly suppressed. But if we provoke these cells to fire by injecting depolarizing current, they actually show an increased firing response. So we're very interested to understand what underlies this relationship and this dichotomy, so to speak, between this difference in the intrinsic excitability and what the cells are actually putting out in terms of their firing activity. And so when we prepare the brain slices from the mice, we also collect the trunk blood so that we can measure the circulating levels of the gonadal hormones. So in the case of the females, this is progesterone, abbreviated as P4 or estradiol. So first, to go over what we found in the case of progesterone, we found that in the mice that were injected with kainic acid and had long cycles, their progesterone levels were significantly decreased compared to both the saline injected controls, as well as the mice that were injected with kainic acid and maintained regular cycles. And then what was interesting, though, was that we saw this relationship on both diestrus and estrus. In the case of estradiol, which is abbreviated as E2, we didn't see any difference between the groups when we measured estradiol on diestrus. But when we did these measurements on the samples obtained on estrus, we saw that the samples from both of the kainic acid injected groups showed elevated levels of estradiol compared to the saline injected controls, which also had slightly lower levels of estradiol compared to what we had seen in the corresponding samples obtained on diestrus. And we didn't see that difference in the kainic acid injected groups. So we're not only seeing changes in the level of the activity and the excitability of GnRH neurons, but potentially also seeing changes in the circulating hormone levels as well. And so to summarize this portion of the studies, for those days of the cycle that we have looked at these relationships so far of estrus and diestrus, we found that the GnRH neurons recorded on estrus from the kainic acid injected groups typically showed strongly suppressed levels of activity compared to the corresponding saline injected controls. And we think that this is primarily due to this increase in circulating estradiol, so an excess of estradiol negative feedback on the system that's suppressing the firing activity of the GnRH neurons. But when we look at the case of diestrus, we see that the cells in the Ka long group show strongly elevated levels of firing. And we think that this is reflected in the decrease in the circulating progesterone levels, which results in a loss of progesterone negative feedback, but only in this group. And so that's why only these cells show the elevated levels of firing. An advantage of having a transgenic mouse with labeled GnRH neurons is that we can do these same experiments now in slices obtained from male mice, which of course don't have a cycle. So we only have one kainic acid injected group here because we're not going to break it down based on long or regular estrus cycles. And what we found in terms of the first looking at the firing output of the GnRH neurons was if we looked at all of the cells combined, we didn't see a significant difference in the mean firing rate of the cells obtained from kainic acid injected mice compared with those injected with control saline. But once again, if we took the data and subdivided it based on the location of the cell that was recorded, we found that actually cells that were located within the medial septum showed much higher rates of firing in the kainic acid injected group compared to the corresponding controls, whereas the cells in the POA and the AHA seemed essentially unaffected in terms of the firing output. And then when we also looked at the intrinsic excitability of these cells, once again, we saw that cells in the medial septum from the kainic acid injected group showed elevated levels of firing or higher evoked firing rates in response to depolarizing current injection. And here we also saw this relationship in cells that had their cell bodies in the POA as well. But once again, we did not see this effect in the cells recorded from the AHA. And so this suggests that as we had seen in females, the cells in the medial septum in the POA are sort of prone to develop increased intrinsic excitability. But again, we have some sex differences in terms of what that means in terms of the firing output. And so to come back to our working model, we'd also looked at the testosterone levels in the males, and we saw essentially no difference at all between the saline injected group and the kainic acid injected group. And so in terms of what we were seeing, it seems like the data from the males is most akin to what we had seen on diestrus in the females, where some of the GnRH neurons show increased intrinsic excitability, but probably because there is no change in circulating testosterone, by and large, there's intact testosterone negative feedback on this system that helps to act as a brake and hold the levels of firing of most of the GnRH neurons down near the control levels. Of course, that's not the case for the cells within the medial septum. And so we are definitely interested in following up to understand what is it about the cells in the medial septum that makes them so susceptible in both sexes to these changes in firing activity, and how that's different from the populations of GnRH neurons in the other parts of the hypothalamus. So as I mentioned, all of the recordings I showed you on the last several slides were done at 2 months after injection. And so, we also see that, at least in the females, we see altered circulating hormone levels. And so that sort of starts to produce a bit of a chicken and egg problem, because it's, well, are we seeing changes in hormones because the GnRH neuron activity is altered, or is the GnRH neuron activity altered because the hormones are altered? So this is where it gets very complicated when you're working in these feedback loops. So we decided to do an experiment of recording from GnRH neurons at just 1 month after kainic acid injection. And we chose this time point because epileptogenesis, or the process of developing epilepsy, is complete in this model by this time. But as a reminder, this was a time point where the estrus cycles in the females were still regular. And in fact, at least for doing the recordings on diestrus, what we found is if we looked at the firing rates of cells in the kainic acid injected mice compared with the corresponding controls, now we don't see that there is the difference in the firing rate. And so it does seem that this presents somewhere between 1 month and 2 months after injection. And so at least it allows us to start to narrow down a time window to understand the time course of these changes. So that'll be a follow-up study. And I just wanted to wrap up with our latest study in this domain. So we had seen that the changes in the firing activity of GnRH neurons changed with the cycle, or the relationship of firing activity in GnRH neurons changed with the estrus cycle. So that led us to also wonder, well, is the epilepsy itself changing with the cycle? Do we see changes in the seizure activity in this model of epilepsy with differences in cycle stage? And so to do these studies, we set up a 24/7, so chronic hippocampal depth electroencephalography, or EEG, which basically means that we have animals that have electrodes implanted into their hippocampus, and then they're connected by these tethers up to what's called a commutator, which allows us to collect the electrical signals, transmit them to a computer through a digitizer. And because they're tethered in this way to the commutator, it allows the mice to roam freely in their home cage. We're also video recording the mice through these webcams, and we have these red lights that turn on after hours so that we can also do the video recording for 24 hours a day as well. And we have this set up so that we can record multiple mice simultaneously. And these are the types of signatures in the EEG that we see that are reflective of seizure activity. So in this study, again, we started with animals that were at least 42 days old. These are all females because we wanted to look at the relationship to the cycle. And so again, we made sure that they had regular estrus cycles for at least 2 weeks, and then performed the same saline or kainic acid injection as we had done before. But now, 2 weeks later, we implanted EEG electrodes into the hippocampus. And we performed 2 sessions of EEG recording. The first was at 1 month after injection, and the second was at 2 months after injection. And during both of these, the mice were also undergoing the daily estrus cycle monitoring. And we found that the exposure to this tethering itself is a bit disruptive to the cycle. And so we did not end up analyzing the EEG data from 1 month beyond just using it to confirm that the mice injected with kainic acid did develop epilepsy by 1 month. But the data that we analyzed was all from the 2 month time point at which that impact of the tethering on the cycle had fully abated. And so what this allowed us to do was to compare the seizure activity within the same mice across different stages of the cycle. And what we found was that if we looked at the percentage of time the mice were spending in seizure activity and used that value recorded on diestrus as the baseline value for each mouse, when we compared that to the values obtained on proestrus and estrus, and we ended up combining those stages because we didn't see differences between the two of them, we saw that there was an increase in the percentage of time spent in seizures on proestrus and estrus compared with the corresponding diestrus baseline. And we also saw this increase in the duration of seizures on proestrus and estrus as well. But I think that, again, you can appreciate that there's quite a heterogeneity here in terms of the severity of epilepsy. You know, some of these mice were showing hardly any time spent in seizures, and then others were showing quite a bit of time in seizures. So we were interested to know whether this relationship of the severity or the burden of the seizures in association with the cycle stage was also related to the severity of the epilepsy itself. So we did these correlational analyses to look at the relationship in the change in the percentage of time in seizures. So taking the value that was recorded on diestrus and subtracting that from the value recorded on proestrus and estrus within each mouse, and looking at it in relationship to the baseline seizure burden on diestrus. So first, the percentage of time in seizures and the seizures per hour, or the seizure frequency. And in both cases, we saw this positive correlation where the mice that showed the more severe epilepsy also showed the largest degree of change in the percentage of time in seizures with cycle stage. And so, of course, we're very interested to understand why there is this relationship. And we think that this will be an interesting feature to follow up on going forward. But I also just wanted to emphasize that we had these mice that sort of clumped down here at very low levels of seizure activity. And frankly, this is not really expected for IHKA mice. It is a model of severe epilepsy. And so since this seemed to be a subpopulation of the mice, we decided to do a follow-up analysis where we looked at only those mice that showed at least 1% of time in seizures on diestrus. And so we said that these were the mice that had the severe epilepsy that's typical of this model. And now when we did the same analysis, we saw, again, strong shifts in the percentage of time in seizures and seizure duration, with those parameters being higher on proestrus and estrus. And now we were also able to see an increase in the seizure frequency as well, with more seizures on proestrus and estrus. And so towards the earlier part of the talk, I talked about catamenial epilepsy, or fluctuations in seizures at different points of the menstrual cycle. And that typically you see these exacerbations in seizures either near the time of ovulation or leading up to the time of menstruation in women. And I'd just like to point out that the same stages that we see the increased seizure activity in our mouse model of epilepsy are rather similar in terms of, again, seeing elevated estradiol and then also potentially these fluctuations in progesterone levels as well. And so, of course, I would never say that a rodent model could recapitulate all features of catamenial epilepsy, but we're certainly interested to use this model to see if it might provide some new targets and ideas of things that might be precipitating these fluctuations with the cycle. Because although I'm sure that these ovarian steroids do play a major role, at the same time, clinical trials that have been designed and have rational bases based on these changes in ovarian steroids, by and large, have not really worked out all that well. And so I really think that, again, it's an opportunity to think a little bit outside the box, and that maybe it's not just about these ovarian steroids, but there could be other features within the hippocampus or within other brain areas that help to influence whether the seizures show fluctuations across these different points of the cycle. And so with that, I'd just like to acknowledge my lab, and especially I'd like to acknowledge Zhang Li. She's a former grad student in the lab. She finished her PhD just about exactly a year ago. And the work I showed you was the bulk of her dissertation. And now we have our current lab members that are also working on related projects, again, looking at the pituitary, looking at synaptic inputs to GnRH neurons, looking at intrinsic ionic currents in GnRH neurons. So we're definitely tackling this at many different levels. Like to, of course, acknowledge the funding, including a graduate fellowship to Zhang that she received from the Beckman Institute, and various grants that have helped to support both this work and other projects in the lab. And with that, I'd also like to thank you for your attention. And once again, thanks to Jeff and Patty and the Beckman team for inviting me to present this work to you today.
All right. Well, thank you so much, Catherine. Virtual applause for a really wonderful and intricate talk. I mean, you can really appreciate the complexity of the brain and all the factors that go into whatever's happening, and especially as we saw, dealing with sex differences in biology is like very, very important and nuanced kind of undertaking. So I believe we should have the participants unmuted, or I can hit that button if I still need to. So first of all, let me open the floor for people to ask questions, in person.
Thought I'd stop sharing the screen so then I can actually see people if they decide.
Sure. Yeah, that's great. Yes. Ask all. So I will, let's see, I've hit the button that says ask all to unmute. So I think people can unmute themselves now. If you can't, put it in the chat so I can do something. Oh, there we go. Allow participants. There we go. Participants, you may unmute yourselves, and so we can just open the floor, and as I see people unmuting themselves, I will call on you, or you can just speak up. And I haven't seen any questions in the chat, so let me just scan the audience here. Oh, good. But I see that Sepida is in the audience, and that reminds me to say that Professor Sepida Sadagiani from psychology is giving our next Beckman Director Seminar on March 4th. She's also a neuroscientist, and she does cognitive neuroscience with humans and looking at network connectivity using both MRI and EEG. So there'll be some interesting touch points between today's seminar and next month's. Okay, does any, Aaron is unmuting himself. So Aaron, do you have a question?
Yes.
Thank you.
So I work with Dr. Huseman, and so we have some work in this area, and both in epilepsy and in healthy in the menstrual cycle. So, and this is mostly because I don't understand all the science and I'm just the imager, can you speak a little bit about what's so special about the hippocampus and the temporal lobe that hormones affect those regions in particular as opposed to the rest of the brain? Because it's, for me, simplistically, it would seem like it should affect the whole brain equally, but obviously it doesn't. So hope you could comment on that.
Yeah, well, hormones definitely affect more than just the hippocampus. I mean, there really are effects of these hormones throughout the brain, but the hippocampus really is sort of particularly sensitive because you see a really high density of receptors for these hormones in both neurons and glial cells there, and that can also change in the epileptic brain as well. So for example, and you mentioned just even in controls, if we look at rodent studies, there are, hundreds and hundreds of rodent studies on effects of estradiol changing synaptic function within the hippocampus, and primarily in ways that promote excitability. So increased a number of excitatory synapses, also decreasing synaptic inhibition. Also, as I mentioned, progesterone, when it reaches the brain, it can be broken down into or converted into other substances called neurosteroids. And these are able to bind to GABA A receptors, which again are prevalent throughout the brain, but especially the receptors that are particularly neurosteroid sensitive, that they have a configuration that allows them to preferentially respond to neurosteroids. They're also seen at very high density in the hippocampus, particularly in the dentate gyrus. So the hippocampus is, and then when you look at the circuit basis of how it's put together, that it's especially prone for these types of network interactions that can lend to seizure activity if sort of given the wrong circumstances, so to speak. But then when you add in the fact that it has this density of hormone receptors and ability to respond in various ways to hormone feedback, it's sort of a perfect storm of a brain region that is prone to have a relationship between epilepsy and seizures. But that being said, I do want to point out, and I'm sure that Graham would say the same thing if he were here, that TLE is, or catamenial epilepsy is not confined to TLE. You can also see patients with cortical epilepsies that show catamenial patterns. So, so that's a bit, it is, the catamenial epilepsy part of it is definitely a more broad spectrum aspect to epilepsy than just within TLE specifically. It's just that our mouse model is a model of TLE.
Yes, it is less common outside of, well, TLE is very common, and it's the catamenial, catamenial issues outside of non-temporal epilepsy cases seem less frequent, at least from my discussions with him. So yeah, it kind of makes sense.
Right, less frequent, but not impossible, let's say.
Right, exactly. And then one more self-serving question is, have you looked into, we have a new 9.4 Tesla animal MRI, and I don't know if you've talked to Bick or Shreyan Majumdar, who's the scientist at 9.4. But
I've heard a lot about the system. I haven't had any official conversations with anyone yet. I think that it would definitely be an interesting thing for us to try to incorporate in our studies. I think if we wanted to try to do EEG recordings at the same time, that might get tricky because of the magnet. but I think that there may be some ways to incorporate that. So yeah, now that that's here, it's sort of will certainly open up some potential avenues of new ways to look at this.
Well, Aaron and Sebeda, I believe that we have done simultaneous EEG and MRI for humans. I don't know if we've ever thought about it for the little guys in the animal magnet.
Yes, we're doing it a ton with humans on the Prisma.
Yeah, so Catherine, we certainly have a precedent for MRI-compatible EEG stuff that we can do simultaneous imaging for, which leads me to my next question, and I guess last question for now, which is, what's the possibilities of MR spectroscopy being helpful for your research agenda? Because we're, again, Jipei Liang and Fan Lam from ECE and BioE have this software package that we've run on humans, and we're hoping and we're planning to use it on the 7T when we start our first study in a few months, hopefully there, to look at different chemicals in the brain. And so how does this intersect with, if you could have a magical way to do MR spectroscopy or other kinds of live spectroscopy on your mouse model, what would you get out of that?
Yeah, so I will answer that question. I think I'll just come back to the EEG for one point, just to say that, yes, I'm aware of human EEG and being able to do that together with the MRI. I guess I'm concerned about the particular electrodes that we use for our mice, and that we would maybe need to modify that a little bit. But certainly, I think having that expertise for human EEG on campus would certainly make that a lot easier for us looking at the little mice. In terms of spectroscopy, I think that's where I sort of think about that a lot now. Is this question of what may be different in the brains of the mice that are more prone to these fluctuations and seizures with the cycle versus those that are just less susceptible. And I think that would be a really interesting thing, whether it's spectroscopy or gene sequencing or just trying to understand like biomarkers that are linked with that propensity. And I think that, again, I sort of, in biology, we see so much heterogeneity, and it leads people to think like, oh, it's dirty. You have to do so many samples to get a good outcome. But I think that here it really helps to emphasize, hopefully, the clinical validity of the model. that, again, it's not that all patients with epilepsy all have the same endocrine problems. And certainly not all women with epilepsy show catamenial seizure patterning or have the relationship of, seizures near ovulation versus those near menstruation. There's a huge spectrum here. And I think that, as much as we can use mice to maybe identify new biomarkers for those things, that's where I could see that type of technology really helping us out.
Yeah, that's a great point. I mean, as one philosopher has said, almost all facts are statistical facts. That there's rarely a sort of all or nothing thing, and so there is a lot of nuance to all these things. Well, let me close out by saying thank you so much again, Professor Christian Hinman, for being here with us today, and thanks to our audience for being here as well. It was a wonderful talk, and thank you so much. And we look forward to seeing everybody back here on March 4th for our next director seminar with Professor Sepida Sadagiani. Thank you so much, everybody.
Thank you.